Hello friends, welcome back to in my YouTube channel, that is Biology Center. Today I am going to teach you protein. Okay. Now in the previous lecture I have stated that in a cellular pool different kind of biomolecules are found. Some are called micromolecules and some are called macromolecules. What are those? Let's see. That is protein, carbohydrate, nucleic acid and lipid. These are the four macromolecules which are found in cellular pool. Okay. Now, but when you're talking about a protein, then it contribute about to, that is 10 to 15% of cellular pool. Please do remember, protein contribute about to 10 to 15% in a cellular pool. Okay. Now, if our body used as a protein as a substrate for the energy, okay, then it will release or yield the energy that is 4 calorie per gram. Please do remember, if our body used protein as a substrate for the energy source, okay, then it will release 4 calorie per gram. Okay. Now, protein is the linear chain of protein is the polymer of, please do remember, protein is the polymer of different kind of amino acid. Different kind of amino acid. Okay, let's see. And they are joined by peptide bond. That is peptide bond. Okay, now let's see how the peptide bond are formed. For example, as we all know that, let's see the amino acid. That is now let's see two amino acid. Then there is a hydration and condensation reaction occur. Okay, in which water molecules are removed. This type of reaction is called condensation or dehydration reaction occur. Okay, this is how. Now the peptide bond is represented by, that is now it becomes dipeptide. Please do remember, it becomes dipeptide and the peptide bond is represented by, that is peptide bond is represented by that is CO NH bond. Okay, now this peptide bond is represented by CO NH bond. Okay, so let's see. When talking about a dipeptide, it means that that is, it indicates that it contains two amino acid plus one peptide bond. Please do remember, in the same fashion, when talking about a octapeptide, then it will simply represent octa, that is, it contains 8 amino acid plus 7 peptide bond. Okay, now when you're talking about a nth peptide, when talking about a protein, that in protein contain n number of amino acids. So you have to learn that is nth amino acid. The protein contain nth amino acid plus n minus 1 peptide bond. Okay, now polymer of amino acid. Many amino acid polymerizes to form protein. Okay, there are 20 standard amino acid are found in a protein, due to which by simply applying permutation and combination, we can make different kind of protein. So there are thousands, more than thousands of protein are found in our body. Okay, that's why protein is called most biodiversity. Protein are most biodiversity, most biodiversity biomolecules. Okay, let's see. Protein are found in, talking about a protein, then protein are found in plasma membrane. Protein are also found in receptor. It is also found in enzymes. It is also found in antibody. Okay, it is also found in antigen. Okay, but when you're talking about a collagen, the collagen is the most abundant protein. Please do remember, collagen is the most abundant protein which are found in animal world. Animal world. Okay, now, but when you're talking about Rubisco, please do remember, Rubisco, Rubisco is the most abundant protein which are found in whole universe, whole biosphere. Please do remember, Rubisco are the most abundant protein which are found in whole biosphere. Okay, now let's see, proceed further. Now let's see a structure of protein. Okay, now a structure of telling way is totally different in a different field. Okay, see, structure of telling way is different in different field. Okay, when you're talking about inorganic chemistry, for example, let's see here, inorganic chemistry, then simply you have to apply the formula. Okay, that is NaCl, MgCl2, etc. Okay, but when we come to organic chemistry, for example, it is organic chemistry, then we have to talking about 2D. Okay, now 2D example is, that is benzene, naphthalene, etc. Okay, now when you come to a physics, okay, then the structure is defined into 3D. Okay, that is physics. It will be talking about a 3D, that is x, y, z plane, x, y and z plane. But when we come to a biology, let's see here, biology, the structure of telling is totally different. Okay, it telling about 4 different levels, that is primary, secondary, tertiary and quaternary structure. These are the four structure which we have to read. Okay, these are the four structure, that is primary, secondary, tertiary and quaternary structure. Okay, now first of all we have to read primary structure. Primary structure of protein. Okay, now it is the linear chain of amino acid. Okay, it is linear chain of amino acid which polymerizes to form protein. Okay, which polymerizes to form protein. Okay, now let's see here. For example, amino acid are linked by peptide bond. Okay, now when talking about a first amino acid, then it is free and it contains NH2 group in it. That is called left end, and in the right end there is a free COOH group. Okay, that is called right end. Okay, so the amino acid are arranged in the fashion of, that is, N terminal to C terminal. Okay, the sequencing of amino acid, you to remember the sequencing of amino acid is given by Sanger's method. Okay, so please do remember. Now, primary structure is more unstable, most unstable. Please do remember, primary structure are most unstable. It is also water insoluble. Water insoluble. That's why it is biologically inactive. Biologically, it is inactive. Okay, now, but when you're talking about a secondary structure of protein, okay, now that is secondary structure of protein. Then it is present in two forms. Okay, let's see here. First form is called alpha helix, and the second one is called beta plated sheets. Beta plated sheets. Okay, when the primary structure, that is present linearly, when it coil upon itself, okay, and fold just like a helix. Let's see here. Now this structure is called alpha helix. For example, that is found in keratin. Okay, now but when talking about beta plated sheets, then for example, that is silk fiber. Okay, now in a single turn of alpha helix, it contain 3.6 amino acid. Okay, now the secondary structure is more stable than protein. Why? Because it contains hydrogen bond in it. So the hydrogen bond takes place between the first one amino acid of NH2 group, then the second amino acid of CO group, NH group and the CO group. It is one amino acid to another amino acid. Okay, hydrogen bond takes place between one amino acid of NH group and the second amino acid of CO group. Okay, let's see. For example, when you're talking about the nth amino acid, then it will make the hydrogen bond between nth plus 4 amino acid. Please do remember, this is the general formula of hydrogen bond. Okay, now let's see. For example, when you're talking about the nth, when you talk, let us consider first amino acid. Okay, now it will make the hydrogen bond between, that is first plus 4, that is fifth amino acid. In the same fashion, talking about a second amino acid, then it will make the hydrogen bond between sixth amino acid. In the same fashion, I am talking about the eighth amino acid. Let's see here. Then it will make the hydrogen bond between 12th amino acid. Okay, now this is how the hydrogen bond stabilize the secondary structure. Okay, now let's proceed further for the beta plated sheets. Now, beta plated sheets also divide into two categories. Okay, beta plated sheets. Okay, also divided into two categories. Okay, first of all, if the two chain of primary structure are running side by side, for example, let's see, that is N terminal to C terminal, and the sequence are same, N terminal to C terminal, and hydrogen bond stabilize this structure. Now, this kind of protein is called parallel plate beta plated parallel beta plated sheets. Okay, parallel beta plated sheets. And opposite to this, okay, if the one stand, that is N terminal to C terminal, are running side by side, and the second have the polarity, that is C terminal to N terminal, and the structure are stabilized by hydrogen bond. Now, this kind of protein are called anti-parallel beta plated sheets. Okay, now come to tertiary structure of protein. Tertiary structure of protein. Okay, now when the primary structure coiled upon itself and form a hazard-like structure, okay, that is just like a hollow woolen ball like a structure, okay, in which crevices, pocket, loops are created, okay, then the structure is called tertiary structure. Okay, tertiary structure show extensive folding. Is to remember, tertiary structure show extensive folding. Why it's so extensive folding? Because it due to the presence of different kind of R group, interaction of different kind of R group. Okay, for example, different kind of force are created, that is ionic force, it may be dipole interaction, okay, it may be van der Waals force, okay, it may be electrostatic force. So different kind of force are created, due to which it's so extensive folding. Just let's see here, that is N terminal to so. Now this is the structure of tertiary structure of protein. Okay, in in which crevices, let's see, all these are in which crevices, pocket and loops are created. Crevices, pocket or loops are created, and this is very necessary for the active side of protein. Okay, it provides activeness, due to which substrate can bind, and it will, and the protein will catalyze the reaction. Okay, now it is the active side which is substrate band. Substrate can bind. Okay, now a tertiary structure is called most biologically active site. This is called most biologically active form of protein. Okay, when we see this structure, then it is like a hollow woolen ball like a structure. Okay, hollow woolen ball like a structure. Okay, now let's see the example. For example, tertiary structure are found in myosin. Please do remember, tertiary structure are found in myosin. Okay, now let's come to quaternary structure. Quaternary structure of protein. Okay, now let's see. When the two subunits of protein combine together to form a quaternary structure. Okay, two subunits. Please do remember, two subunits combined together to form quaternary structure. Okay, let's see. For example, it may be a single protein, that is primary structure, it may be secondary structure, it may be two secondary structure, it may be two tertiary structure, it may be one tertiary structure and one secondary structure. This is how the quaternary structure are formed. Okay, let's see. Now this is the quaternary structure. One is alpha helix and second one is tertiary structure. Okay, for example, that is hemoglobin. Please do remember, hemoglobin contains 2 alpha plus 2 beta chain. That is 2 alpha plus 2 beta. Now this is how the protein structure are formed. Okay,